Why rice water makes soil explode with life. The microbe science most gardeners never learn. Researchers culturing microbial samples from fermented rice water found bacterial concentrations measuring between 100 million and 1 billion colony-forming units per milliliter. To put that number in context, a standard probiotic supplement marketed to support human gut health typically contains between 1 and 10 billion colony-forming units per entire capsule. A single milliliter of properly fermented rice water delivers a microbial payload approaching that same threshold, 1 milliliter, a fraction of a teaspoon. And when that concentration is diluted into a watering solution and applied to garden soil, researchers measuring the response found detectable changes in soil microbial diversity within 72 hours of a single application. The farming communities of rural Japan, Korea, and Southeast Asia have been fermenting rice water and applying it to their garden plots and paddy fields for somewhere between 300 and 500 years. They called it by different names in different regions and prepared it with slight regional variations, but the core practice was identical everywhere it appeared. Wash the rice. Collect the cloudy wash water. Allow it to ferment for a specific period under specific conditions. Dilute it. Apply it to the soil or directly to plant foliage. Observe the result. Pass the observation forward. They were not doing microbiology. They were doing something more reliable than microbiology at that moment in history. They were running a multi-generational field trial with consistent methodology and observable outcomes, and the outcome they kept observing was the same. Soil that received fermented rice water produced more vigorous plants, recovered faster from stress, and maintained productivity across seasons with less external input. The experiment ran for centuries before any laboratory existed that could explain the mechanism. Modern soil science is now explaining the mechanism, and what it is finding is that the traditional practice was not approximately correct. It was precisely correct, almost as if someone had designed it with knowledge of microbiology that did not exist yet. To understand why fermented rice water does what it does to soil, you need to understand the problem it is solving. And to understand that problem, you need to spend some time inside a structure that most gardeners have never thought about at this level of detail. Think of the top 6 inches of your garden soil as a living city, not a simple one. A complex, layered, interdependent metropolitan system with distinct infrastructure sectors, specialized worker populations, and critical supply chains that must remain intact for the system to function. This city does not run on electricity or fuel, it runs on carbon. Specifically on the flow of carbon-rich organic compounds through a series of biological processing stages, and every stage of that processing is managed by a different category of organism. When you apply fermented rice water to soil, you are not adding a nutrient. You are adding workers, specialized workers, to 3 specific departments of this city that are chronically understaffed in most home gardens. Understanding which departments and why they matter is what separates a gardener who uses rice water because they heard it works from a gardener who uses it because they understand exactly what it is doing at the cellular level. The first department is the city's primary energy grid. This grid is managed by a class of bacteria called lactobacillus, and in healthy soil, these organisms occupy a structural role so fundamental that everything else in the biological city depends on them to function. Lactobacillus species are lactic acid bacteria, meaning their primary metabolic output is lactic acid produced through the fermentation of sugars. In the context of soil, this is not incidental. It is architecturally critical. The lactic acid they produce does 3 things simultaneously that benefit the entire soil ecosystem. First, it acts as a biological pH buffer. Lactic acid is a weak organic acid, meaning it releases hydrogen ions gradually rather than all at once, and it does so in a way that resists sharp swings in soil pH. Most beneficial soil microorganisms operate within a relatively narrow pH range of roughly 6.0 to 7. When soil pH drifts significantly outside that range, microbial populations crash because the enzymatic chemistry that drives their metabolism stops working correctly. Lactobacillus populations, by continuously producing and consuming lactic acid in the decomposition process, help stabilize the soil chemical environment within the range where the entire biological city can function. Second, lactic acid bacteria produce compounds with antimicrobial specificity, meaning they suppress certain categories of pathogenic and parasitic soil organisms without harming the beneficial populations. Researchers studying the soil microbiome have found that soils with high lactobacillus density consistently show lower concentrations of fungal pathogens from genera including Fusarium and Pythium, both of which are responsible for the root rot and damping off, conditions that kill seedlings and weaken established plants. The mechanism is competitive exclusion combined with direct chemical suppression. When beneficial bacteria occupy the available physical space in the soil pore structure and produce compounds that interfere with pathogen metabolism, pathogens cannot establish at population levels sufficient to cause damage. Third, and most consequentially for the gardener, lactic acid bacteria are primary decomposers of plant-derived starches and simple sugars, and the byproducts of their decomposition activity are direct inputs to the nitrogen and phosphorus cycling systems that plant roots depend on. They are the first link in the nutrient processing chain. Without a robust lactobacillus population, the chain slows at its origin point and everything downstream slows with it. What this means for your garden is that applying fermented rice water is functionally equivalent to hiring thousands of new workers for the city's energy grid, workers that immediately begin producing the chemical outputs on which every other organism in the soil depends. The second department of the underground city that fermented rice water directly supports is its photosynthesis bypass system. This is a category of organisms called phototrophic bacteria, and their presence in traditional agricultural systems was not understood mechanistically until relatively recently, which makes their inclusion in a centuries-old farming practice all the more remarkable. Phototrophic bacteria, particularly purple, non-sulfur bacteria from the family Rhodospelaceae, are capable of using light energy to drive metabolic processes that would otherwise require oxygen or organic carbon inputs. They occupy a unique ecological niche in the soil because they can operate under conditions, low oxygen, low light penetration, moderate moisture, that are hostile to most other beneficial organisms. Their contribution to the soil system is the production of bioactive compounds including amino acids, nucleic acids, and biotin that function as direct growth stimulants for the plant root system. Soil science shows that plants growing in soil with active phototrophic bacterial populations demonstrate measurably higher rates of root hair development, which directly increases the surface area available for nutrient absorption. A plant with a more extensive root hair network is not just slightly better at absorbing nutrients. It is structurally better positioned to support a more extensive mycorrhizal fungal association, which then multiplies the nutrient access advantage further across the entire root zone. Phototrophic bacteria also fix atmospheric nitrogen independently of the more commonly discussed Rhizobium species. They do so through a slightly different enzymatic pathway, but the end product is the same. Ammonium that plant roots can absorb without any synthetic input. In a soil with a healthy and diverse bacterial community that includes both rhizobium and phototrophic nitrogen fixers operating simultaneously, the total biological nitrogen production can be substantial enough to significantly reduce or, in well-established traditional garden systems, eliminate the nitrogen deficit that drives most fertilizer purchases. What this means for your garden is that the phototrophic bacterial fraction in fermented rice water is adding a secondary nitrogen-fixing population to your soil that operates in the microenvironments where your primary fixers cannot reach, filling biological gaps in the system that most gardeners never knew existed. The third department receiving direct support from fermented rice water is the city's Construction and Structural Engineering Division. This is managed by yeasts, specifically wild yeast strains from the genus Saccharomyces and related species that colonize the surface of rice grains during the fermentation process. Yeasts are not commonly discussed in soil biology conversations oriented toward home gardeners, and that absence in mainstream gardening advice represents a significant oversight, because what yeasts produce as metabolic byproducts has consequences for soil physical structure that no mechanical amendment fully replicates. Yeasts produce a class of compounds called biosurfactants. These are molecules with a hydrophilic end that attracts water and a hydrophobic end that interacts with organic particles and mineral surfaces. In soil, biosurfactants function as natural wetting agents that dramatically improve the distribution of water through the soil profile. Compacted or hydrophobic soil, the kind that forms when organic matter is depleted and the surface becomes waxy and repellent to water, resists infiltration in ways that cause water to run off rather than penetrate. Biosurfactants produced by yeast activity in the soil break this hydrophobicity by lowering the surface tension of water at the soil-particle interface, allowing water to move into previously impenetrable zones of the soil profile. This is not a trivial benefit. Water access is the rate-limiting factor in microbial metabolism across the entire soil biological community. Without adequate moisture distribution, bacterial populations cannot sustain the enzymatic activity that drives nutrient cycling. Fungal hyphae cannot extend into dry zones to build the network. Earthworms, which require moisture to move through soil and to maintain the mucus layer that lines their tunnels, retreat or die in chronically dry microenvironments. Every biological system in the soil city is moisture dependent, and the yeast-produced biosurfactants in fermented rice water directly improve the moisture distribution that all of them require. Yeasts also produce B vitamins, particularly thiamine and riboflavin, as metabolic byproducts, and these compounds function as cofactors for the enzymatic systems of the bacterial population sharing the same soil environment. This is the underground city operating as an integrated economy rather than a collection of separate processes. The yeasts produce vitamins that accelerate bacterial enzyme function. The bacteria produce lactic acid that stabilizes the pH environment where yeast can survive. The phototrophic bacteria produce amino acids that feed both. Remove any one group and the productivity of all three declines. Introduce all three simultaneously as a properly fermented rice water preparation does, and you are seeding a complete internally cooperative biological community rather than adding a single organism in isolation. What this means for your garden is that the yeast fraction of fermented rice water is solving a water distribution problem and a soil chemistry problem at the same time, without any mechanical intervention and without any synthetic chemistry. Now understand what killed these three departments in the first place, because this is where the modern gardening oversight becomes visible with uncomfortable clarity. Imagine you are the city administrator, and someone informs you that the water supply to the engineering district has been contaminated with a broad-spectrum industrial solvent, not a targeted one. A broad-spectrum one that does not discriminate between the organisms you want dead and the ones the city depends on. This is the situation that unfolds in soil biology every time a synthetic chemical with antimicrobial or antifungal properties reaches the soil surface, whether applied intentionally or arriving as runoff. These categories of products are designed to eliminate biological activity in the target pest or pathogen. They succeed. But the soil does not have a firewall that stops the effect at the target organism. The disruption spreads laterally through the biological community because the chemistry that drives pathogen metabolism and the chemistry that drives beneficial organism metabolism share enough common features that a broad-spectrum disruption affects both. This is not stated to condemn the use of all synthetic inputs under all circumstances. It is stated to explain the mechanism by which soil biology degrades in the average home garden over time, and why that degradation feels invisible until the productivity losses become impossible to ignore. Standard garden center guidance does not typically walk through this mechanism. The step of explaining what a chemical application does to the non-target biological population is almost always skipped. That is a common oversight with compounding consequences. Because every season the biology remains depleted is another season during which the self-renewing system that traditional communities maintain for generations is not doing the work it is capable of doing. Now bring the traditional conformation into full resolution, because the ancestral practices around fermented rice water reveal a level of observational precision that is genuinely humbling when viewed through the lens of modern biology. The traditional preparation method used by farming communities across and Southeast Asia was not random. It was the product of iterative refinement across generations, and each specific parameter of the preparation turns out to be biologically meaningful. Traditional practitioners fermented the rice wash water at ambient temperature for a period of between 3 and 7 days, stopping the fermentation when the liquid developed a specific sour smell and a slightly cloudy, milky appearance. They were not using pH meters or bacterial density measurements. They were using sensory indicators that, it turns out, accurately reflect the state of the microbial population inside the liquid. The sour smell is the lactic acid output of the lactobacillus population reaching significant concentration. The cloudiness is the combined visual effect of bacterial cell bodies, yeast cells, and their metabolic byproducts suspended in the liquid. Traditional farmers knew that a preparation that smelled too sharp or showed unusual coloration had gone wrong and would not produce the desired effect. They were quality controlling a living biological product using nothing but their senses, and they were doing it accurately enough that the practice produced consistent results across hundreds of years of application. The dilution ratio that traditional communities settled on is equally precise in its biological logic. Most traditional preparations were diluted between 1 part fermented liquid to 20 parts water and 1 part to 30 parts water before application. Soil science offers a clear explanation for why this range was optimal. At full concentration, the lactic acid content of properly fermented rice water is high enough to temporarily depress soil pH beyond the tolerance range of some beneficial organisms. At dilutions more extreme than 1 to 30, the microbial concentration per unit of applied water drops low enough that soil establishment of the introduced organisms becomes unreliable. The traditional dilution window, arrived at through observation of plant response rather than laboratory measurement, sits precisely in the range where pH impact is buffered and microbial delivery remains effective. The timing of application within the growing season also reflects accumulated biological understanding. Traditional practitioners applied fermented rice water at the beginning of the planting season before seeds or transplants went in, again during the period of most active vegetative growth, and once more at the transition into fruiting. These application moments correspond in biological terms to 3 distinct phases of soil microbial demand. Pre-planting inoculation establishes the foundational bacterial community before root systems develop and begin making demands of it. Mid-vegetative application supports the rapid expansion of the mycorrhizal network that coincides with the plant's maximum growth rate. Fruiting stage application supports the lactobacillus-driven nutrient cycling activity that needs to peak precisely when the plant is diverting maximum energy into seed and fruit development. Traditional farmers tracked these moments by plant appearance and seasonal observation. The biology they were responding to was real and consistent, even if the vocabulary for it did not yet exist. The closed-loop dimension of this traditional system is perhaps its most elegant feature. Rice is a primary food crop in the regions where this practice originated. Rice washing produces rice water as an automatic byproduct of food preparation. Fermenting that water requires no additional inputs, no purchased organisms, no specialized equipment. The clay pots or ceramic vessels traditionally used for fermentation provided an imperfect but adequate seal that allowed the natural wild yeast and bacteria present on the rice surface and in the ambient environment to colonize the liquid without requiring inoculation from an external source. The organisms needed for the fermentation were already present. The substrate they needed was produced as a food preparation byproduct. The application vessel was a clay pot or a section of bamboo. The total cost was the labor of collection and the patience of waiting. And the return was a soil biological management system sophisticated enough to maintain productivity across generations without soil degradation. This is the model that modern, sustainable agriculture is attempting to replicate through expensive commercial preparations, proprietary microbial blends, and branded soil probiotic products. The ancestral communities had the core version of it for centuries at a cost that approached zero. So here is how you bring this into your own garden, and it is important to be specific about the method because the preparation details are where the biology is either preserved or destroyed. Use the wash water from rinsing raw, unprocessed, non-enriched white or brown rice. Enriched rice has synthetic additions on the grain surface that can interfere with the fermentation process. Wash approximately 1 cup of rice in 2 cups of room temperature water, agitating it for 30 to 60 seconds to dislodge the surface starch and the naturally occurring wild yeast and bacterial populations from the grain surface. The wash water should be distinctly cloudy. Drain off this water and reserve it. This is your fermentation substrate. Place the cloudy rice wash water in a glass jar or ceramic container. Cover it loosely with a cloth or an imperfect lid that allows gas exchange but prevents contamination from insects or debris. Allow it to ferment at room temperature, ideally between 65 and 75 degrees Fahrenheit, for 3 to 5 days. In warmer conditions, fermentation proceeds faster. In cooler conditions, it may take the full 5 to 7 days. Begin checking at the 3-day mark. The preparation is ready when it smells pleasantly sour, similar to plain yogurt or a mild vinegar, and the liquid has taken on a slightly milky opacity. If it develops an unpleasant putrid smell rather than a clean sour smell, The fermentation has been colonized by the wrong organisms, and the batch should be discarded. When the fermentation reaches the correct stage, dilute it at a ratio of 1 part fermented rice water to 20 parts clean water. Apply this diluted solution directly to the soil surface around your plants rather than onto the foliage, particularly during the initial applications, to direct the microbial payload into the root zone where it will do the most work. Apply approximately 1 to 2 cups of diluted solution per square foot of garden bed. Do not oversaturate. The goal is to introduce the organisms into the soil moisture film where they will colonize, not to flood the root zone in a way that creates anaerobic conditions hostile to the aerobic bacteria in the community. If you are starting with severely depleted soil, begin conservatively. Apply to a small section of your garden first and observe plant response over 2 to 3 weeks before treating your entire growing area. In healthy or moderately depleted soil, many gardeners observe visible improvements in leaf color and plant vigor within 2 to 3 weeks of the first application. In severely depleted soil, the initial microbial population may need 1 to 2 months to establish at densities sufficient to produce visible surface effects, because the organisms need to multiply to working population levels before their collective metabolic output becomes significant enough to change plant nutrition at a measurable scale. Apply every 2 to 3 weeks through the growing season for ongoing support, reducing to once monthly in cooler conditions when microbial metabolism slows. Store prepared fermented rice water in a sealed container in the refrigerator for up to 1 week after fermentation is complete. Beyond 1 week, the lactic concentration continues to build and the pH of the liquid drops to levels that may stress the organisms themselves. The science behind this practice is no longer theoretical. Soil biology research has characterized the organisms involved, measured their metabolic outputs, and documented the soil response to their introduction. The mechanism is understood at the cellular level. The benefit is quantifiable. And yet, the knowledge that this practice exists, that it has existed for centuries, that it requires no purchase and no specialized knowledge beyond what the traditional farming communities preserved and transmitted, is not a standard part of the conversation in most garden centers or mainstream gardening media. That is not a conspiracy. It is simply the result of a system oriented around products meeting a market that never learned to ask for something that cannot be packaged and priced. The ancestral farmers who developed this practice were not withholding it. They were using it openly as part of the daily rhythm of their food culture. The rice water came from the rice they were washing for dinner. The fermentation happened on the kitchen shelf. The application was as routine as watering. It was embedded so completely in the ordinary texture of daily agricultural life that it required no special designation as a technique. It was simply what you did with the water from the rice. You have it now. The biology, the mechanism, the preparation, the application, and the historical confirmation across 3 centuries of traditional practice. The rice water is not magic. It is microbiology. And the organisms it delivers to your soil have been waiting for exactly this kind of invitation. Give them the substrate, give them the time, then step back and let the city